Hello students, this is your Prasanna ma'am from NEET World. Today we are here to discuss the most difficult segment of the questionnaire, that is assertion reason questions and statement based questions from the chapter cell cycle and cell division. So if you look into the first question children, wherein in assertion it is given like interface occupies 95% of duration of cell cycle, it is correct, and in reason it is said like interface is the long non-dividing phase, reason is also correct. But here reason is not explaining on why interface is occupying 95% of duration of cell cycle. As this is not explaining, reason is not explaining assertion, it will fall into what option ma? It will fall into option B, where both assertion and reason are true, but reason is not explaining assertion. So before ending this question, I would like to give you a gist on cell cycle. Cell cycle has two phases. What are they? Interface and M phase. Interface and M phase. Wherein interface is also called as preparatory phase or resting phase. Interface is also called as preparatory phase or resting phase, and interface is divided into G1 phase, S phase and G2 phase. M phase is the cell division phase. M phase is what beta? Cell division phase, and M phase is further divided into prophase, metaphase, anaphase, telophase, and then cytokinesis takes place, cytokinesis is followed by karyokinesis. Karyokinesis has 4 substages. What are they? Prophase, metaphase, anaphase and telophase. So karyokinesis is followed by cytokinesis in the M phase. In which phase beta? M phase. So keep this in your mind. This is very basic flow where you need to understand and fix this in your mind. You can answer many questions if you keep that in your mind. Okay. Assertion. Karyokinesis follows cytokinesis. No. Here only you can see, right? Cytokinesis is following karyokinesis. M phase is divided into 2 phases. In one phase nuclear division happens, in the other phase cytoplasmic division happens. Once the nuclear division is done, then only cytoplasmic division takes place. Here cytokinesis follows karyokinesis. Karyokinesis is not following cytokinesis. Assertion is wrong. Cytokinesis is division of cytoplasm. Reason statement is true. So when assertion is wrong and reason is true, our option will be what ma? Option will be D. Okay. Next, assertion. G1 phase is the interval between mitosis and initiation of DNA replication. Yes, G1 is the phase between mitosis. Yes, it is in between M phase and DNA replication phase. DNA replication phase is nothing but S phase, that is synthetic phase. Yes, this statement is true. Next, the cell is metabolically inactive in G1 phase. Wrong. Cell is metabolically active in G1 phase, and most of the cell organelle duplication happens in G1 phase. So in that case, when reason is wrong and assertion is true, our option will be what ma? Option is C. Okay. Next question, if you see. Yes. So here during mitotic anaphase, centromere of each chromosome splits and chromatid separate. During anaphase what happens ma? During metaphase, if you see, all the chromosomes are aligned on the equatorial plate or metaphasic plate. Here the spindle fibers are attached to the kinetocores and they are pulled towards the pole. In anaphase what happens? Centromere splitting takes place and chromatids will move with different shapes to the opposite poles. Yes, this is correct. During anaphase, chromatids move to the opposite poles. Both the statements are true, but reason is not explaining assertion. Then the option will be B. Okay. Next question. Small disc shaped structures at the surface of centromeres are called as kinetochores, small disc-shaped structures at the site of, if this is the centromere, small disc-shaped structures at the sides of centromere to which the spindle fibers are attached. These disc-shaped structures are what? They are called as what, Nana? They are called as kinetochores. They are called as kinetochores. So, yes, this is correct. Next, kinetochores serve as the site of attachment of spindle fibers to the centromeres. Yes, they are attaching the spindle fibers to the centromeres. Correct, both assertion and reason are true, but then it is not explaining assertion. Here what will be your option? Option B is the correct answer. Next question, if you take, here in assertion it is given like prophase is the first stage of mitosis. Prophase is the first stage of mitosis which follows S phase and G1 phases of interphase. No, if you look into that again, interphase is divided into what, beta? G1, S and G2. Then in metaphase, prophase comes, right? The first stage of metaphase is what? Prophase. So, this is following what? S phase, then G2 phase, then it is prophase, then it is prophase. So, here assertion is wrong. If you look into the reason, prophase is marked by initiation of condensation of chromosomal material. Reason is correct. When assertion is false and reason is true, then our answer will be C. Option what, beta? Option C. Next, assertion. Cell growth results in disturbing the ratio between nucleus and cytoplasm. True. Whenever the nuclear division is happening, the very first in the prophase, the very first stage of karyokinesis, what happens is in a cell, the nuclear membrane gets degenerated, nucleolus get degenerated, organelles get degenerated, and the cytoplasm will get very much thick and dense. Cytoplasm gets what, beta? Cytoplasm gets very much thick and dense. In that dense cytoplasm, the chromosomes are free to undergo the karyokinesis process. Here the nuclear division or genetic material division easily happens. Nuclear division easily happens as the nuclear membrane, nucleolus and organelles got disintegrated into the cytoplasm. Cytoplasm gets dense and inside that chromosomal division happens easily, wherein they can move to the opposite poles in the anaphase, and then in telophase again the last step of karyokinesis, the nuclear membrane, nucleolus, they get reappeared and chromosome gets uncoiled to what, ma? Chromatin material during telophase. So, here cell growth results in disturbing of nucleus and cytoplasmic ratio, 100% correct. Mitosis helps the cell to restore the nucleocytoplasmic ratio. True. So, here what will be the option? Option B is the correct answer, where both assertion and reason are true and reason is explaining the assertion. The process of pairing of chromosomes is called as synapsis. Correct. Synapsis occurs during leptotin. Wrong. During prophase 1 of meiosis, we have 5 substages. What are they, beta? Leptotin, zygotine, pachytene, diplotin and diakinesis, of which pairing occurs in zygotine stage of prophase 1 in meiosis 1, not in leptotin stage. So, here assertion is true and reason is false. Our option will be C. Correct? Next question, beta. Here, crossing over leads to recombination of genetic material into 2 chromosomes. Correct. Crossing over leads to what, ma? Recombination of genetic material on the 2 chromosomes. Crossing over is the exchange of the genetic material between 2 homologous chromosomes. We have already discussed what are homologous chromosomes, but then for your recalling, again I am telling you, homologous chromosomes are 2 chromosomes which has 2 alleles of a single gene, We know what is a gene. Gene is a functional unit of inheritance. Let's take a character. Each gene will code for one character or trait. Let us take like some tallness. Let us take tall. The two different alleles of tall, dominant is capital T, recessive is small t. Two alleles located at the same loci on two different chromosomes, then these two chromosomes are called as homologous chromosomes. These two chromosomes are called as homologous chromosomes, wherein two different alleles of the same gene are located at the same loci. These two chromosomes are brought together during zygotine stage and they are called as synaptonemal complex, and it is called as what ma? Synaptonemal complex. During Pakitine stage, these two alleles get crossover, and here the crossover takes place with the enzyme recombinase. Crossover occurs with the enzyme recombinase. Okay, so here two alleles which are located on the same loci of two different chromosomes, then two different chromosomes, as two alleles are present on the same loci, they are called as homologous chromosomes. Exchange of genetic material takes place in the homologous chromosomes. So crossover occurs between two homologous chromosomes. This is true. When both A and R are true, then the option is B. Next, in assertion, if you see, the crossing over is an enzyme mediated process. Yes, we just discussed the enzyme is what beta? Recombinase. The enzyme involved in crossing over is lyase. No, it is not lyase, it is recombinase. So yes, here A is true and R is false. Then the option is C. Okay. Next. Yes, here the final stage of meiotic prophase 1 is diplotin. Wrong. The final stage of meiotic prophase 1 is diakinesis. It is what ma? Diakinesis, wherein terminalization of chiasmata occurs. What happens? Terminalization of chiasmata takes place. Okay. Next, the beginning of diplotin is recognized by dissolution of synaptonemal complex. True. Okay, the beginning of diplotin stage occurs by dissolution of synaptonemal complex and followed by formation of chiasmata, that is X-shaped chromosomes. That is what ma? X-shaped chromosomes. Yes. So yes, reason is true and assertion is false, then our option will be C. Okay. Next, the stage between two meiotic divisions is called as interkinesis. Interkinesis is generally short-lived. Of course, two statements are correct, but here in reason there is nowhere the explanation of interkinesis is given, then our option will be B. Okay. Next, metaphase 2 begins with splitting of centromere of each chromosome into two. Wrong. Splitting of centromere occurs only in the anaphase 1, only in the anaphase 2. Sorry, it occurs only in the anaphase 2 of meiosis. Anaphase 2 of meiosis. In anaphase 1, what happens? Two set of chromosomes will be present. Two homologous chromosomes will be present and it forms bimetaphasic plate during the cell division. In metaphase 1 what happens ma? Bimetaphasic plate is formed. Bimetaphasic plate is formed wherein during anaphase 1 there is no centromere splitting. There is no centromere splitting, wherein of this pair of homologous chromosome, one pair goes to one pole and the other pair goes to the other side of the cell. Okay. Yes. So here in anaphase 1 there is no centromere splitting. In anaphase 2, of course in the metaphase 1 only one metaphasic plate or equatorial plate is seen, and in anaphase 1 same like in mitosis centromere splitting takes place. So here assertion is wrong. In anaphase 2 chromosomes align at the equator, of course this is correct. Now here if you see assertion is false, reason is true, then what is your answer? Answer is C. Next, assertion variations are important for the process of evolution, correct. Next, meiosis increases the genetic variability in the population of organisms from one generation to the next, correct. Okay, from one generation to the next, meiosis increases the genetic variability, but nowhere here the variation concept is explained in the reason, then our option will be B. Okay, next question. Yes, next we have entered into statement based questions. A cell cycle includes both interphase and M phase. Okay, pro, meta, ana and telophase includes only M phase. Includes what beta? Only M phase. G1, SG2 includes only interphase. Karyokinesis and cytokinesis includes only M phase. So it includes both interphase and M phase. Interphase includes G1, S, G2. M phase includes karyokinesis and cytokinesis Karyokinesis is further divided into prophase, metaphase, anaphase and telophase. Next, read the following statements about cell division and select the correct ones. M phase represents the phase when actual cell division occurs, and it is the phase that represents the phase between, and one phase represents the phase between two successive M phases. This is what interphase. This is what interphase. Interphase represents the phase between two successive M phases. Yes, one is true. He is saying that in M phase actual cell division occurs, true, and in interphase, that is a phase between two successive M phases, he is saying true. In 24 hours average duration of cell cycle of a human cell, cell division proper lasts for about an hour, correct. M phase constitutes more than 95% of duration, wrong. What constitutes more than 95% of duration of a cell? That is interphase. That is what interphase. Okay. So here what is the option? Which two statements are correct? Which correct ones he is asking? Whenever you go through a statement question, look whether he is asking you to identify correct statement or incorrect statement, then only you look into the options. Okay. So here the correct statements are option 1 and option 2, then it is A. Next, select the incorrect statement regarding S phase of interphase. It occurs between G1 and G2, correct. DNA replication in the nucleus in this phase, yes. Centriole duplication in the cytoplasm, true. As DNA doubles, number of chromosome doubles, wrong. Actually, you will see no chromosome appearance only in S phase. Chromosome appearance starts in the metaphase. Complete condensation of chromosome completes in the metaphase, begins in the prophase. In S phase, you will not see any chromosome number. But then to make you clear, only DNA content doubles but not the chromosome number. He has said you this. So, for example, if this is the number of chromosome in G1 phase. In G1 phase, you have how many chromosomes? 2 chromosomes. 2 chromosomes here. Okay. In S phase, of course chromosomal duplication happens, DNA duplication happens, but then here also chromosome number is 2 chromosomes only. Chromosome number is 2 chromosomes only. You will identify the chromosome number with centromere number. Okay, you will identify the chromosome number with what centromere number. Here also it is 2 chromosomes, here also it is 2 chromosomes. But here if you take 2C is the amount of DNA, here it will be 4C amount of DNA. Okay, DNA content has doubled, but then chromosome number remains same, chromosome number is not increasing. So yes, as DNA is double, number of chromosome also get doubles. This statement is wrong. Here only you identify before choosing. He asked us to identify incorrect statement, then our answer will be D. Next question. Which of the following cannot be found during the study of cell cycle? The diploid cells found in G0 and G1 phase? Yes, we can identify whether diploid cells are present or haploid cells present. Cells with twice the normal DNA content, cells with twice the normal DNA content in the early M phase. Next option, cells with chromosome number doubled in S phase. See again, just by looking into this term, you can easily identify chromosome number will never double in S phase beta, only DNA content gets doubled. Just by looking into that, you can identify option C is the answer, because this never happens in the cell cycle. Next question. Read the given statements and select the correct option. Statement 1 is saying that at the end of prophase, cells do not show Golgi complexes, endoplasmic reticulum and nucleolus, correct? The complete disintegration of nuclear envelope marks the end of the first phase of mitosis. What is the first phase of mitosis? Prophase only, right? It is prophase. So yes, this is also true. Both statement 1 and statement 2 are correct. It is option A. When they reappear, they reappear in telophase. Next, which of the following is the key event of anaphase of mitotic division? Which of the following is key event of anaphase of mitotic division? Chromosomes are moved to spindle equator and get aligned through spindle fibers to both poles. Wrong. This happens during which phase? This happens during metaphase. Centromeres split and chromatids separate. Correct? Chromosomes cluster at the opposite spindle poles and their identity is lost as discrete elements. This happens in telophase. This happens in which phase? Telophase. Okay, wherein chromosome will lose their identity and they will get uncoiled to what? Chromatin in telophase, and nuclear membrane, nucleolus, everything is reappeared. Okay, yes, so here the option will be B. What is true about telophase stage in mitosis? Chromosomes lose their identity as discrete elements. In telophase he is asking. Yes, A is correct. Chromosomes cluster at the opposite spindle poles. True. Nuclear envelope, nucleolus, Golgi complex and ER gets reformed. True. So, all these statements are correct with respect to telophase. Next question. Read the following statements and identify the correct option he is saying. He is asking us to identify the correct option. Yes. So, statement 1, plant cytokinesis occurs by cell plate method. True. In plant cells, furrow gradually deepens and divides cell cytoplasm into two. Wrong. So, in plant cell what happens ma? In plant cell, first in middle side cell plate is formed. In middle what is formed? Cell plate is formed. Cell plate is formed with the help of middle lamellae that is secreted from the Golgi bodies, and this cell plate will move towards the periphery, dividing the cell into two equal halves. Okay, this moves towards the periphery and this conducts the process of cytokinesis in plant cell. Such type of division is centrifugal. Such type of division is what beta? It is centrifugal, occurs in a plant cell. If you look over here, cell, animal cell. If you look into an animal cell, cell deepens inside from the side and it makes a formation of a furrow. It deepens inside and formation of a furrow takes place. So here from the periphery it deepens inside. Such pattern of cell division is centripetal. Such pattern of cell division is centripetal that happens in animal cell. Okay. So here the statement 2 is wrong and statement 1 is true. Then our answer will be B. Okay. Next. Yes, read the given statements and select the correct option. Crossing over is the exchange of genetic material between two homologous chromosomes, correct? Next, crossing over is an enzyme mediated process and the enzyme is recombinase. Enzyme is what ma? Recombinase. Next. So here two statements are correct. Two statements are correct, then both statement 1 and statement 2 are correct over here. Next question. Select the incorrect statement. Kinetochores serve as the sites of attachment of spindle fibers. True. During anaphase, centromeres split and chromatids separate. Correct. Nucleolus, Golgi bodies reform during telophase. True. Mitosis usually results in the production of 4 haploid daughter cells. Wrong. Mitosis always produce 2 daughter cells. Mitosis always produce 2 daughter cells and these daughter cells are identical to parent cells. These daughter cells are identical to parent cells. Clear beta? So yes, statement D is wrong and here they asked us to identify the incorrect statement. So our option will be D. D is the correct answer. So with this we have finished or we have covered up the assertion, reason and statement based question models from the chapter cell cycle and cell division. In the next class we will discuss about TSEAPCET previous year questions and NEET previous year questions of this chapter. Thank you.